Our next talk will be by Lisa Gazdzinski. She's a research associate in the Branch Lab at the Hospital for Sick Children in Toronto. Dr. Gazdzinski's work focuses on deepening our understanding of the effects of traumatic brain injury on neural networks and behavior using mouse models and advanced MRI and histological methods. Her talk today is titled Disrupting Oligodendrogenesis during development impacts myelin and axons. Please take it away, Dr. Gazdzinski.
Okay, thank you. Does everyone see my screen okay?
That's great.
Okay, great. So thanks to the hosts for planning this and putting it together. It's been a really interesting couple of days. It's a little bit different, the stuff I'm going to talk about here, because I'm not going to look at neurodegeneration, but rather development. Oops, I'm not in the right place, am I? Let me see. So as we've been reminded many times, myelin is the fatty substance that surrounds axons, and it's an extension of oligodendrocytes. And it's really important for the timing of axonal propagation and signals throughout the brain. And brain regions don't act in isolation. It's really important that they act in a coordinated fashion. And so this speed is important. So white matter development happens over a long period of time. The brain at birth doesn't have very much myelin at all, and this myelination increases really rapidly in the first few years of life, peaks at around, the rate peaks around 5 years of age, but it continues at a lower rate for decades in the human. So relevant for this particular study that I'm going to tell you about, in the mouse brain, myelination is happening, of course, also over a long period of time. But what happens, or what's important here, is that different white matter tracts, and this happens in humans too, but different white matter tracts myelinate at different times. And so in this particular study, the sort of relevant ages that we're looking at are about 3 weeks of age to maybe 7 weeks of age. And we're looking at 3 different white matter tracts. The optic tract tends to myelinate quite early, and so it's sort of finished by about 3 to 5 weeks of age. The anterior commissure starts a little bit later and continues for a little bit longer, and then the corpus callosum continues much longer into adulthood in the mouse. So in our lab, we're interested in the effects of disrupting white matter maturation during development. So one of the sort of conditions that we look at a lot in our lab is traumatic brain injury. But this idea can kind of be applicable to any sort of process that can disrupt white matter maturation. So if we think of the sort of black line as the normal white matter maturation or maturity level over age, an insult early on can maybe lead to a premature maturation of the white matter. Perhaps it could be delayed, or maybe it could be a mixture of the two and just be disrupted in some way. And any of these could have implications for behavior or brain function in general. So the objective of the current study that I'm going to tell you about was to characterize the effects of disrupting oligodendrogenesis, or disrupting the development of myelin during development, on white matter microstructure and behavior in mice. So in our lab, as in many of yours, we look at white matter structure at different scales. So we can think of like a macroscopic scale where we look at volumes of different structures, or perhaps the shape of different structures. Microstructure can be looked at using histological methods and electron microscopy methods. And like all of you, we're trying to use MRI to get a sense of the microstructure, but at the whole brain level, which, as we've seen, we can do with histology, but it would be much nicer if we didn't have to do it in as complex of a way as needed for using histology. So in this study, we use an inducible genetic mouse model in which the MERV gene is conditionally knocked out in oligodendrocyte precursor cells. So myelinating oligodendrocytes arise from oligodendrocyte precursor cells, which mature into pre-myelinating oligodendrocytes, and then with this transcription factor are able to mature into mature myelinating oligodendrocytes. So in this mouse model, which is they've got the MERV gene floxed in NG2 producing or expressing cells, so OPCs, under the inducible CRE. And so through the administration of tamoxifen, we can knock out this gene in these OPCs, and therefore they can no longer mature into oligodendrocytes. And so these mice cannot make new myelin, but they do keep whatever myelin was there already. So it's not a demyelinating model. So the design of this experiment, mice were given tamoxifen at either 3 or 5 weeks of age. So 3 weeks of age in a mouse is about the time, it's the day that we wean them. So it's about sort of young toddler age. 5 weeks of old is, 5 weeks of age is sort of the onset of adolescence. So in these mice, the CRE positive mice are ones that will be affected by this tamoxifen and will have the oligodendrocyte maturation halted, and the CRE negative mice should not have that happen. So they were injected at 3 or 5 weeks of age, and then 2 weeks later, they underwent some behavioral testing. So we put them through the Y-maze test and also did some fear conditioning. They were tested for fear memory the next day, and then they were perfusion fixed for either ex vivo MRI or electron microscopy. And so our MRI protocol is here. It's a 7 Tesla scanner. It was all ex vivo, a 3-shell protocol at 100 micron isotropic resolution, and we processed it with DTI and NADI, and also used the B0 images to get structure volumes. So here are the volume results. So for all of the plots I'm going to show, the blue are the CRE negative mice, so sort of like the wild type, you can think of them. The pink are like the CRE positive mice that would be affected by tamoxifen. The ages here are the age at which the tamoxifen was administered. And so I put these little bars down here to remind you that the imaging time point is 2 weeks after the tamoxifen injection. So these mice here are actually 5 weeks at the when they're scanned, and these ones are 7 weeks when they're scanned. For all of the data, I analyzed the ages separately with a mixed effects model where we were looking at the covariant for genotype, the CRE status, sex, and with a random effect for litter. So in the tracts that are, if you look at the blue, the sort of CRE negative mice first, you can see sort of what would be the normal maturation of these tracts, or the normal brain, like relative volume of the tracts changed over the course. So in tracts that are sort of mostly myelinated, like the optic tract, we really don't see much increase from the young to the older mice. Same thing with the anterior commissure, because it was sort of complete by 5 weeks. But in the corpus callosum, you can see that we have this increase in volume. And then in the CRE positive mice, we can see that for the younger mice, where we would have been affecting the myelination at the, during a peak time of myelination in the anterior commissure and the corpus callosum, we see that there's a very significant reduction in the volume of those tracts in the younger mice that we don't see in the optic tracts. So this is a really nice readout, and it's nice to see that we see kind of what we would expect to see if you stop myelination. But it doesn't get it all at the microstructure. And so we were looking to use diffusion methods to get a better understanding of what the microstructure is looking like. And unfortunately, when we did this with DTI and NADI, we really didn't see any significant differences with CRE status at any of the ages for any of the metrics. And so this was quite perplexing to us. I mean, as all of you know, you sort of assume that the diffusivity would be quite sensitive to the myelination, but it's basically that either the metrics had trend toward the opposite direction of what you might expect, or there was really just no readout from the diffusion data. And so this really prompted us to dig deeper into this, and so we have turned to electron microscopy to try to understand why the diffusion would be so insensitive to what would likely be a pretty significant effect actually in the tissue. So here are some example images from the Cre-negative and Cre-positive mice, just in the corpus callosum. And so we're looking at the myelinated axons in here using a program called Axon DeepSeg. And so we're working with a group in Montreal who's developed this machine learning segmentation for the electron microscopy images. So it segments the myelin and the axons, and so we can get morphometrics out of these images for the myelinated axons. And then we're working with that group to also have it be able to identify the unmyelinated axons, but at this point it's not quite ready for that. So we have counts of the unmyelinated axons that we've done manually. And so the sort of most striking effect with like the axon counts in general, and this is still preliminary, we're not finished all of the counting for all of the images that we have, but it looks like the younger mice that were Cre-positive have more axons in the images, so a higher axon density, but that most of them, or a bigger proportion of them, are made up of the unmyelinated axons. And I guess this makes sense. You would expect this. If you stopped myelination, you would have more of them that just end up being unmyelinated. So that makes sense. And I think that, the increased density that we have here is likely just shows that as myelination has not really happened, the axons haven't been pushed apart so much, and you just end up with a higher density in a smaller volume. But if you look at the myelinated axons separately, where you can get measures of the G ratio and the axon diameter from the morphometrics, we see that we have a trend level decrease in G ratio in these young mice, which could either come from a smaller axonal diameter or thicker myelin. And just, again, this is preliminary, but it looks like it's coming from, at least in this corpus callosum data, that these axons are smaller than you would expect them to be, even though they were already myelinated at the time where we halted the myelination. So just a quick look at the behavior as well, because we also saw some behavioral effects. So the Y-maze test is kind of a classic test of working memory. So the mice are put in this Y, and then they're allowed to explore it. And mice are sort of naturally exploratory, and so basically they're expected to go into, say arm A and then B, and then go back to C before they go back into A. So they kind of have to remember where were they just, and they go to the one that they haven't been to most recently. And that's called a successful alternation. And so for the younger mice, the Cre-positive mice actually looked like they performed better in this task, which was a little bit unexpected, but also the idea of better is a bit maybe misleading, because it's hard to know sort of if they were supposed to be, at this kind of level and they're actually higher. They sort of are behaving more like the older mice. So it's almost like a premature behavior. And then similarly in the contextual fear conditioning, we saw this difference in the younger Cre-positive mice, where they were freezing less than would be expected for their age, and they're looking more like the older mice. So basically, to conclude, when we stop oligodendrogenesis during development, it leads to a decreased myelin content in the tracts that were later to mature, and we detect this as smaller volume in those structures. But there's indirect effects that are happening on to the axons themselves. probably changes in axon diameter and density that are really confounding the interpretation of the diffusion MRI metrics that we've looked at, so that it doesn't seem to reflect the myelination differences that we would have expected. And so we really do, as we're advocating for in this entire conference, that we really need more complex models or more comprehensive protocols that combine different methods of looking at the tissue to pull apart the changes in myelination and axonal properties, particularly during development or pathology. And for this particular study, these mice with these halted oligodendrogenesis at a young age seem to have altered performance in memory tasks. So with that, I'd like to thank the people in the lab, the people who have done most of the work on this study are Koster Gamarathas. He did all of the EM acquisition, and Miranda and Jordan have done the behavioral testing. Thank the people at Axon, working on Axon DeepSeg in Montreal, and the scientists at the Mouse Imaging Center for helping with the scan protocol. And thank you for listening.
Thank you very much. Very interesting. I look forward to seeing how that work develops. Well, we're just a little bit behind schedule, so maybe, Sajad, just one question from the chat for now, and then we save the rest for discussion.
Okay, so we have one question from Dean Chen. Thanks for the talk. Thinking about clinical importance of these for humans, are these conditions in humans that may stop proper myelination in early childhood?
So I get this question a lot, and my answer is just like, I'm sure there is. It wasn't sort of the motivation for this work at this time. Definitely traumatic brain injury, as we have studied, does have effects on white matter microstructure that looks similar in some ways to this, so that we're seeing kind of a premature white matter maturation in some of our work with traumatic brain injury in development. But I will have to, I'm sure that exists. I'm sure there's lots of different pathologies that kind of involve the disruption like this of oligodendrogenesis, but I don't know any others right off the top of my head.
Okay, thank you so much. Yeah, in the interest of time, we can move to the next.
Okay, thank you.
Thank you. Thanks again.